Hi everyone, it's Mr. Stratton. Today I want to talk to you about proteins. Looking at that molecule to the side there, it's actually really pretty, but its beauty is actually very complicated. Okay, our essential questions for proteins, always with all of our biomolecules, how do the structures of those molecules relate to their functions? Well, we're also going to add an extra layer. How does a change in the amino acid sequence change the structure and the function of that protein? So proteins have a wide variety of functions. In fact, being coded for by your DNA, they're the way that your genes are expressed. So we produce things like enzymes for digestion, your immune proteins like antibodies, plenty of hormones like insulin, structure of your body like collagen, and transport like hemoglobin in your blood that transports oxygen. The monomer is amino acids, and the polymer, we call them polypeptides or just proteins. The individual amino acids have a very stable, simple backbone: nitrogen, carbon, carbon. The nitrogen group is the amino group on one end. The carboxylic acid group on the other end provides the acid part of the amino acid. And in the center, that central carbon atom with a hydrogen and R. Now, there is no R on the periodic table. R simply stands for residue, and there are a bunch of different possible things you could put there. For us, as humans, we actually have 20 amino acids. I've grouped them into hydrophilic and hydrophobic amino acids. So the hydrophilic ones can be polar, like the ones in the top box there. The ones at the bottom would be the ones that are acidic or basic, so they're actually charged. They're either positive or negative. And then the hydrophobic amino acids on the other side, which have, if you look at their R group, it's really just containing primarily carbon. There may be some other more electronegative atoms in there, like a sulfur or a nitrogen, but in reality, they're buried deep within the molecule, and so the electrons are going to be pulled equally, and so those molecules are not going to have a pole to them. Whereas look back at the hydrophilic ones, and you'll see that they've all got oxygens on their surface, or sulfur, or amino groups, or carboxylic acid groups, and the result is those are actually going to really like water. Put them into a protein, and the consequence is the hydrophilic ones are going to want to be near the surface because our bodies are mostly water. So in that aqueous environment, they're going to go to the surface, whereas the hydrophobic ones are going to hide deep in the middle of the protein. And so it's going to have consequences for the structure of that protein. So out of the 20 amino acids that we use, we can produce 11. So the other 9 are called essential amino acids, and they have to come from your food. So if you're a vegetarian, it means that you have to consider that when you're designing your diet. I will say this: tyrosine does require phenylalanine to be produced, so whether you count that as essential or non-essential, that's up to you. But the meaning is that we don't have enzymes to make the other 9. Okay, amino acids are joined using, surprise, surprise, dehydration synthesis, and broken apart using hydrolysis. So dehydration, of course, removes the water, and it's going to form a particular type of covalent bond. It has a specific name. It's called a peptide bond. Put a bunch of those together, polypeptide. That's a protein. All right, the end that has the amino group is called the N-terminus or amino terminus, and the end of the protein that has the carboxyl group is called the C-terminus or carboxy terminus. The structure of a protein is vital for its function, and in this case, there's 4 levels of structure we're going to get into. But the base level is the amino acid sequence, which is determined by your DNA. So our proteins range from 44 amino acids to over to 27,000 amino acids. So there's a huge range, but the average protein has about 375 amino acids. So that's actually pretty long. If you think of amino acids as words, that would be a pretty long sentence with 375 words. So this is key. The structure of the amino acids chain, right, the proteins, the structure of that protein determines its function. If the structure is wrong, the function's wrong. So there are 4 levels. So from primary, which is just the amino acids in a row; secondary, which is some local folding; tertiary, which is more distant, more complicated 3D structure; to quaternary structure, which is where you have more than one polypeptide chain interacting. So I'm going to go through these individually. The primary structure is just the amino acids in the order that they're in. So that would be just the sentence. What order are the words in to make sense? Okay, so in this case, this chain of amino acids, it's just peptide bond, peptide bond, peptide bond, and that's it. This is the most important level of structure, however, because it determines all the rest of the levels of structure. Because the order of the amino acids is vital for the next layers of folding. The secondary structure is hydrogen bonding, and it's local. It's only amino acids that are close by. So there's two levels of two different forms of secondary structure you need to know. The first is the alpha helix, so it's where it coils back on itself, and the second is beta pleated sheet, which is where you've got pleats like on a skirt where it's folded back on itself. But both of them are stabilized by hydrogen bonds. Now those hydrogen bonds are weak, and these can be broken relatively easy. In fact, they're the first level of structure to be broken. Tertiary structure, this is going to be more distant, and this is where the R groups come into play, because the R groups are going to interact. So this could be hydrogen bonding, it could be ionic or covalent bonding, or it could just be that, hey, these are hydrophobic amino acids and they want to hang out together and get away from water. Now the one that I want to point out right now is the disulfide linkage. That's actually a covalent bond, and specifically it's between two cysteines. So cysteine has this R group here with the sulfhydryl group on it. And when two of those get close together, they form an actual covalent bond that's very strong and stabilizes protein structure. Why is this important? Well, again, the structure of the protein determines its function. And you can see, in some cases, how many cysteine residues people have in a protein called keratin by looking at their hair. Because if you've got curly hair, it's because you have a lot of cysteine residues spelled out in your DNA, which ultimately is in your protein, in the primary structure. And when they interact, they can actually covalently bond to each other and put curls in your hair. Now, I personally do not have a lot of cysteine residues in my hair, but if I wanted to add some add some sulfur, I could. I could go to a shop, they could put some sulfur in my hair, they could make some temporary little sulfur-sulfur bonds in there. That would be a perm. Unless you're Bob Ross, you're not going to pull that off. Now, on the other hand, if you have curly hair and you would like to break those disulfide linkages, you can go, they can put sulfur on your hair and actually get those sulfurs to not bond to each other, but bond to just a free sulfur molecule, and that would be straightening your hair. All right, so we've got the primary structure, which leads to secondary structure, which is the alpha helix and the beta pleated sheet. We've got tertiary structure, where the molecule folds back on itself. And finally, quaternary structure is where you have more than one polypeptide. So in this case, we have two beta hemoglobins and two alpha hemoglobins. And you can see the alpha helices within there. Those little coils are the alpha helix. Alpha helices is plural. So this molecule carries oxygen. This actually has some other stuff in it, including an iron. It has 4 iron atoms buried in there that actually helps to carry the oxygen. So, reviewing this whole thing then. So the primary structure is the order of amino acids. Those then coil back on themselves using hydrogen bonds or pleat themselves using hydrogen bonds to form alpha helix or beta pleated sheet. That then is you're going to have R groups from distant amino acids interacting, forming covalent bonds, ionic bonds, hydrophobic interactions. That's going to be your tertiary structure. And then quaternary structure is more than one polypeptide. So this is vital to know because it's going to come up again and again throughout biology. So here's an example. Insulin actually has 2 polypeptide chains together. There's actually a third one that you produce at the same time, but that gets released and it goes off and it actually activates cells and signals them to do things, called C-peptide. But the A chain and the B chain, they are coiled back. They are coiled. It's not showing you any secondary structure here, but it does show you the disulfide bonds that form. Now, realize that, especially that top one there, it is not forming a bond across that distance. Instead, those are folded back towards each other. So this doesn't really show you the 3D structure. This does, however. So you can see there are alpha helices buried in here. There are disulfide linkages, which are shown as those yellow bonds. And then there's a beta pleated sheet down at the bottom too. So an alpha helix is shown as a coil. A beta pleated sheet is actually shown as an arrow, a flat arrow. So you can see that this 3D structure is vital. This must have this shape in order for insulin to do its job. If it doesn't, well, then the insulin doesn't work. So my wife being a diabetic, we have insulin in our fridge. if my wife were to leave that insulin sitting out, what might happen? Well, it might actually break down. I'll get to that in a minute. All right, so what can proteins do? They can be enzymes. So in this case, lactase is going to cause hydrolysis of lactose. They can be defensive proteins like antibodies, where they can then stick to the outside proteins on something like COVID. They can be storage proteins. This is just a source of amino acids for a baby. Baby's going to drink the casein in milk and then break it down and get the amino acids out. Or a baby chick is going to have the ovoalbumin, which is the white of the egg, and it's going to be able to break that down and release amino acids. Transport proteins. So these are things that are like channels in the cell membrane that allow it to move molecules through. So the yellow part, all those beta-pleated sheets, those there are causing a pore, like a hole, that's specific to certain molecules that come through into the cell. Receptor proteins. So those little blue things on the outside of the cell there, their job is to fit a certain molecule and then cause some sort of change in the cell. In this case, possibly open up a channel protein. Hormones. So this is insulin. Its job is to regulate glucose uptake. So what it does, in this case, it's that little triangle. It's going to bind to another protein, a receptor, which is then going to cause a change in the metabolism of the cell. And then you guys all learned in freshman biology that, hey, the mitochondria is the powerhouse of the cell. Well, did you know your mitochondria have feet and they walk around inside your body? They do. Yeah, there's lots of little motor proteins that when ATP binds to them, it causes them to change shape. And when they change shape, it allows molecules to walk. It's actually how your muscles work. Structural proteins like keratin, or in this case, collagen. So collagen has 3 polypeptide chains. That's its quaternary structure. And it forms these coils that are stretchy, and it's what allows your skin to be stretchy and elastic. Of course, when you get older, your collagen starts breaking down, you stop making as much, and the result is your skin gets wrinkly and saggy. Sorry. Okay, so why is the protein structure so important? Simply because it determines its function, right? Hemoglobin carries oxygen, and if it's not shaped properly, it can kill you. So in this case, normal hemoglobin has a glutamic acid in position number 6, but mutant hemoglobin, sickle cell hemoglobin, has a valine there. The difference between those two is glutamic acid is hydrophilic and valine is hydrophobic. Now, the code for those is in the DNA. So if somebody has a mutation in their DNA, it codes for the wrong amino acid, it makes that protein fold wrong because that valine does not want to be near water. It's going to fold into the middle of the protein. And you see that beta subunit there is the wrong shape. Well, it actually causes the hemoglobin molecules to stick to each other and stretch out into these long chains that then make the cell stretch out into these sickle shapes. And then in the blood vessels, they get caught and they cause clots. So one little tiny change in the DNA leads to the wrong amino acid, makes that protein shape wrong, makes the proteins stick to each other, makes the cell shape wrong, and then causes clots and death. Yeah. There's a lot of consequence to just one mistake in the DNA. Okay, the environment can also change the protein structure. So I told you, my wife keeps her insulin in the fridge. The reason being, she doesn't want it to denature because the hydrogen bonds, the secondary structure, could break. And if it does, then, well, that protein's no good anymore. It has to have the right shape in order to work. So sometimes if the protein has changed shape, there actually is a benefit. So in this case, before you were born, when you were a fetus, you didn't have beta hemoglobin. You had gamma hemoglobin. So you see that graph there? You've got alpha and gamma as a fetus, and then the gamma tails off, and you start producing beta as you're born. The reason being is you don't want your blood cells to have the same affinity, the same strength of attraction for oxygen as your mom's, because you might steal oxygen, your mom might steal oxygen from you. So instead, you want your hemoglobin to be stronger than your mom's. And gamma and alpha together are stronger than beta and alpha together. So the result is you always take from your mom, which is beneficial for the fetus. Another example is your eye color. The color of your eyes is a protein, or multiple proteins actually. And so whatever combination of proteins you produce results in the color of your eye. If you make no protein at all, your eyes are sky blue. If you make lots and lots and lots of really dark protein, your eyes might be black. Most of us are somewhere in between. And so whatever combination of proteins, their structure determines their color, which determines your phenotype, the way that you look. Finally, antibodies. This is important because of COVID. Antibodies help you fight off invading bacteria, viruses, and other things. And you can produce a wide variety of these because the end of that has a little variable region. And then you have multiple variable portions of your genes that allow you to change up the structure. So when an invader comes in, your body is going to keep producing different antibodies until it finds the right one. That response takes some time, which is why you're not immediately immune to something, why it takes time for you to get over a cold, or why when you get a vaccine, it takes a couple weeks for you to really have these ramped up and produced so that you're ready to go and to fight off the virus. So what I want you guys to do is visit this site and play around with a random protein. Put it in water, put it in oil, change the indicator around and see what happens. Check out, just kind of play with it and see what happens when you change things about your protein. Okay, most important aspect is its 3D shape. Why? Explain to me. Okay, mutation results in a polar amino acid being replaced with a nonpolar. What is that going to do to the structure and function of that protein? And finally, viruses mutate frequently. So sometimes they've made a flu vaccine based off what they saw was coming, and by the time it gets here, it's mutated. So why wouldn't you have immunity to that virus anymore? Thanks for watching.